Hello.
Hello.
How are you?
Yeah, I'm fine.
Good.
What about you?
Pretty good.
Yeah.
Yeah, so it's a holiday, I know, in the U.S. So I don't know how many people are going to show up. I know that Karan and Jiahang and Wataru are not in the U.S., so I don't know.
Oh, okay. Yeah.
Okay. So do you have any updates?
Right now, what I'm trying to do is found an algorithm on GitHub which works on neural networking. So it does is it converts the 2D images of an object which are taken from different views into a 3D object. So I'm trying using that algorithm. I'll be training the algorithm and producing a result. Then I'll see what happens for.
All right. Yeah, that sounds
Yeah.
interesting. Yeah. What's the name of
And
the?
also, I,
Oh, what was that?
and also I discussed with Karan about the future progress. Yeah.
Okay.
The algorithm is. I only remember the short form. It's NERF. NERF.
Okay.
I'll share the link in the chat.
All right. Sounds good. All right. All right. Yeah, I can share the link when you're ready. Um, I guess we can get started. I was just waiting for a few more people, but I guess we can do this now. So I wanted to go over some things with the community period. So first of all, welcome to the community period. If you're watching this later, or if you're watching it now, for Google Summer of Code. So our community period actually started last week, and I've been in the Slack with some resources for people. Um, I want to make sure that both projects are covered, but I also have some general materials, and I'll go over what those are. Um, and then, okay, here we go. And then we'll go through some of these other. I have some other topics to cover today, and we'll see who shows up. So this is the NERF link. So this is. Okay, so this is a link to a GitHub repository. And this is the code. So it's got a lot of labels actually. Unity. This is for like a mixed reality or a virtual reality volume rendering sort of thing, and then you can also use it for other things. Yeah.
Yeah, and the examples they have shown is interesting. Like, I think things can work with that.
Okay. Yeah.
Yeah.
This looks good. So this is this is an NVIDIA project or product. Yeah. Okay. I think I've heard of this before. I was just trying to like kind of remember where I'd heard the name. Yeah, NERF. So this is sparse input photos from different angles. So you're taking the same image from different angles. You're building a three-dimensional mesh, and you're getting 360-degree views. Okay. And yeah, this looks really good. So yeah, let's keep us posted on that.
Yeah. Sure.
All right. Hello, Karan.
Hi.
Hi.
Yeah.
Hi.
Yeah, so we discussed this nerve thing, and we kind of went through 2-3 more ways to go about when modeling the project. So we had, I think, this meeting day before yesterday only. So the thing is, we'll be dividing, like, at least like I'll be doing the modeling part, and Harikrishna will handle some of the projecting part, and how to finally display the 3D object on the website. So for that we discussed potential options. So far we'll be most probably going forward with the Hero script only, and I think we till if we can get a better fit for the algorithm that we have to use in this, we'll probably try that. But so far, we'll still continue with the, methods that I had outlined. So that is there. Anything else you want to add, Harikrishna?
No, that's it. I guess those are all the questions.
Yeah. So we'll keep on, checking out new things as well. Like NERF was kind of an interesting, idea that we wanted to investigate as to how it would fit our model here. So that is there.
Okay. That sounds great. Yeah, I'm looking forward to seeing it. Like I said to Harikrishna, I've heard of this before, but I've not really explored it, so be interested to see. Hello, Jiahang.
Oh, hi.
How are you?
Yeah, I'm good. I'm sorry that I could not attend the meeting the last week. Yeah, I'm quite busy this month.
Yeah.
Yeah, we've already finished a paper and we've submitted to another conference which is similar to the Learning on Graph. Yeah.
Okay.
The New Rips 2022. Maybe you've heard that. Yeah.
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 Oh yeah. Well, you don't know if you've gotten accepted yet, but
yeah. Yes, it's too busy this month.
yeah. Well, congratulations. Yeah, that sounds good. All right. Well, that's great. So why don't we get into the... I wanted to go through some community period things for GSoC. So all of you got the messages in the Slack.
Yeah.
Okay, so let me go over some of this. So this is our Slack. This is, well, the OpenWorm Slack. So like I said last week, the community period is about looking through the OpenWorm Foundation more generally and seeing what's there. So if you look through the Slack, you'll see that there are a lot of different channels here. And that just corresponds to all the different things that have been going on in OpenWorm. So there's a lot of stuff going on in OpenWorm besides us. You have like C302, which is a group that models the connectome. And they do a lot of stuff with they have a software where they model the 302 neurons of the C. elegans nervous system. So, they're doing a lot of pull requests in this channel. This is C302 beginners. It'll load the history here. And there's also a place to discuss things about C302. So this is if you're interested in neural networks or graph neural networks or things like that, this is an application to the adult nervous system. They have a formal modeling software, but they're interested more generally in that what they call connectome. And this is a good summary of it here in the GitHub README. So there's a GitHub for OpenWorm, and you don't have to join that, but just to let you know that it exists and that there are a lot of contributions being made there, and that they're doing a lot of things with different aspects of modeling C. elegans. They're doing a lot of things in Python, so we're probably going to do a lot of things here, and we're going to be not contributing directly to the OpenWorm GitHub repo, but it's going to be something that maybe people will be interested in. Then there are other groups like ChannelWorm, where they're modeling ion channels in the cells themselves. So all cells in C. elegans have ion channels. The neurons have ion channels that produce electrical activity that is similar to, it's not exactly an action potential, but it's similar to an action potential. And, so the nervous system is very ion channel dependent, but you also have other cells that have ion channels. It's very important to know what those ion channels are doing to really model what the cell is doing and the behavior of the cells. So that's that channel. We have an education channel, which is somewhat dormant. It's been, but in the past we've had a lot of educational opportunities. We have educational materials for different things. This is the hermaphrodite nervous system in C. elegans. So that's like the 99% of all worms in C. elegans are hermaphrodites. So this is an overview of the nervous system. You'll find a lot of interesting things in this channel. INCF has some training space materials that get repurposed in OpenWorm and things like that. So this is something you might want to check out if you want to know more about like our community and how people are learning about C. elegans. There's also like engineering, which deals with some software engineering issues. OpenWorm really started as sort of it had a heavy emphasis on software engineering because people were trying to engineer all these different pieces of software that are put together to form OpenWorm and form this, where you're simulating different parts of the worm. So there's that aspect of it, but there are also these different systems level aspects. So there's movement analysis, which is where you have people talk about, I think I talked about this last week, where they're doing movement tracking of the worm. So they're looking at a worm through a microscope. They're looking at it moving along this agar surface, and they're kind of characterizing the movements because C. elegans has a number of stereotype movements that you can record. And, if you have the right technology, they use what they call a movement tracker, which is like the cell tracking technology that we've talked about here, but it's just tracking the actual worm and its movements. So they actually have data sets that are available. And those data sets might be of use to you as you work on some of your models and validating them. I'm not so sure about digital microspheres, but definitely the Graph Neural Networks project, you might look into some of these, like the TIRPSY tracker software, and they also have something at movement.openworm.org. And that's a bunch of data sets that have this movement tracking data in them. And it just, it's a way to validate, what the output's supposed to be. It's a way to validate some of the algorithms you're writing. So we also have in the past had a lot of focus on building robots. So there was an early version of an OpenWorm robot that was like a hard robot. It looked like a Roomba vacuum cleaner. It was a round robot that would move around, and was programmed with the algorithms that were developed from the OpenWorm Connectome and other types of software tools that were developed in OpenWorm, and they were able to drive this robot to do different behaviors. And so that got a bit of a media attention, and if you look it up online, you look up OpenWorm plus robot, you'll find a number of articles on this, and a lot of them are pretty spectacular in the sense that they're not really, it's just a robot that you put software into and it behaves in a certain way. It's not like going to become conscious. So, but in any case, that's where the project's been. More recently, they've worked on building soft robots that have, and I know Tom Porteges, who's been in this group a number of times, or he's been in this group historically. He hasn't really attended meetings much anymore, but he's done work with robotics as well. So, he's really good at like building robots and building software for like artificial intelligence, adaptive systems type applications. So, this is something that, you have a worm, it behaves in a phenotype, it has a brain. We can simulate the brain, and we can also simulate the body. We can simulate the movement of the body. We can simulate the soft aspects of the phenotype, the cells and the outer cuticle and things like that. So this is another channel you might want to check out. You have to go back quite a ways to find some of the meatier work here, but that's the thing with Slack. Everything is archived. Of course, the problem is it's trying to find interesting things. So there's just a lot of, if you really want to get into the guts of like how people build robots, This has been chronicled in this robots channel. It's really kind of interesting. So that's our Slack. I just wanted to bring that up because I think, I want to encourage you to go outside the bounds of DevoWorm for a couple weeks and see what's out there in our organization. And if you find someone you might find interesting, you want to talk to them, drop them a line, send them an email or send them a Slack message, or I can get you in touch with someone specific if you want to talk to them. It might be better for me to contact them because they know me more than they know you, so they might be more likely to get in touch with me. But there are a lot of different things going on in the OpenWorm Foundation. They vary because it's an open source organization. There's a lot of variability in when people are working on things. So, DevoWorm has been pretty constant. But like robotics, you'll get people who are interested in robotics. They might start working on a little project, and then they might produce something really nice. And then they might drop it for a while, and then come back to it maybe 2 years later. And that's the nature of these sorts of open source activities. And which brings me to my next point, which I'll show you in a bit, which are these readings that I sent to you in the Slack channel. So I'll talk about that in a minute. Hello, Susan. Nice that you could make it today.
Yeah, it's a bit busy here, but
yeah,
I'm taking a break. So.
Okay, good to have you. So yeah, this is the community period, so you're encouraged to explore the bounds of your organization, and your organization is OpenWorm. DevoWorm is within OpenWorm. It's a project. You have other projects doing other things. Our focus is development and the development of the worm. So we have that area that we're interested in, but there are these other areas as well. So next part I'll go to the DevoWorm channel specifically, and I'll talk about some of these links that I put in the chat. So this is a drive of things that they're resources for open source and open source project management readings. So I have this library of things that are based around learning about open source software and how it's made and how to manage it, and then some other resources in open source project management. And so I encourage you to read through these. I'll go through some of them in a minute. And there are other, these other channels like movement analysis and robotics might be useful for you as well. Now, this person here in these two videos that I posted, his name is Stephen Larson, and it's been more than 10 years now. Him and a couple other people started OpenWorm. They got together online and they said, "This is great." They had this idea about Stephen, for example, got his degree in artificial intelligence. He got a master's degree from MIT in artificial intelligence, and then he got a degree in neuroinformatics or neuroscience at UC San Diego in the U.S. And he was finishing that up. And there were a couple other people in Europe working on the Neuron package, which is a package to simulate neurons. And they were working on these different projects. And Stephen, got in touch with them and said, I think he made a call and asked, if people are interested in simulating C. elegans. Now, why would you want to simulate C. elegans? Well, at the time, and it still continues to be a key model organism for a lot of biologists. It has a fixed number of cells in the adult. It has a very distinct and sort of deterministic developmental lineage tree where all the cells, what they're going to become. And it's very well characterized. And so he said, wouldn't it be nice if we could build a digital model of C. elegans? And so everyone was excited, and we started off, and I didn't really actually come into this community until about 2014 or 2015. So it was a while after they started it. But these two videos here are good intros to what OpenWorm is trying to achieve and sort of the mission statement. So this first one is an INCF talk. So this was at INCF. And he was talking to that group about OpenWorm. And this is, the subtitle was A Community Developed In Silico Model of C. elegans. So this was back in 2014. So this is going to be a bit dated. But he talks about some of these different things that they're trying to do at the time. So this is more of their vision for the future. This is good because it lays out a roadmap for what they wanted to achieve, and some of those things have been achieved and some have not. This talk here, this TED Talk, is a little bit different. This is kind of a focus on how the C. elegans, or its a common name is roundworm, is our best bet to unlock the secrets of the brain. So one of the reasons why people want to understand C. elegans, and especially the nervous system of C. elegans, is that it's good to study. It's a very small nervous system. We know every cell. We know the connections between the cells, which is unusual for a nervous system. But it's also, you can tell some things about the human brain or mammalian brains by, looking, especially if you do the molecular biology. You look at the genes being expressed, or you look at some of the genes that exist in, mutated genes that exist in some strains of C. elegans. One of the things C. elegans is known for is this aspect of having these defined mutants. So you can have a C. elegans that has a specific mutation that it always carries. You just let it reproduce and it always carries this mutation. And you can compare it to a wild type, which means that it has like a normal, in the wild sort of genotype. And so it carries a lot of mutations, but they're not what they call defined mutations. So there's just genetic diversity. A lot of these defined mutations have some functional consequence. So if I have a mutation in some gene, it might mean that I don't have a certain neuron, or that I'm move very slowly, or that I don't form a full cuticle, things like that. And that's useful for just kind of interesting to see how mutations work on the phenotype. But it's also very interesting for disease. You can create defined mutants for different neural diseases that you might see in humans or in other mammals. And you can understand things like aging. You can understand other types of processes that go on. So this is something that's very useful. And modeling the worm in this way as almost like a digital twin. They didn't use that terminology back then, but it's something that would be sort of analogous to this, is very useful. And that being said, the digital models that exist now are, they're not complete. They don't really have a genome. They don't have a genotype, but they're actually quite good at the cellular level in terms of understanding what's going on there, having virtual models of the cells and things like that. Then this link here is a link to some materials that we generated during an open house back in 2016. So we had an open house, and it kind of went through a lot of the different projects. So there are materials for each project. There's some background readings. There's a talk, maybe about 5 to 7 minute talk on these different projects, and it kind of recaps sort of the origins of each project. Now it's a bit dated. It's from 2016, so some of these projects have moved on since this time, in terms of its progress. But this is a good, I think, guidepost to some of the things that have been done in OpenWorm. There are two other things I'm going to share with you this week, and I don't know why I didn't do it before, but the first is that there was a special issue of the Royal Society B, which is the Proceedings of the Royal Society B, and that was in 2018. And the OpenWorm Foundation was invited by the Royal Society to give like a seminar in London on OpenWorm. And so there was a lot of excitement around that. And there were a number of talks. Some the people were inside OpenWorm, some were outside. They were experts in the field. And they talked about a lot of things about the connectome and about computational modeling of the worm. And that ended up becoming a special issue of the Royal Society B. So I'm going to send a link to the journal, the special issue. I can get access to the papers if you want them. Actually, I think I have a library of them. I'll send you the library of the papers as well. And then there was some other, there was some like live activity at the workshop that I have a summary of. I'll send you that as well. So that's the one thing, the one link that I wanted to share with you, and that's a little bit more recent update on some of this work. The second link is that there's a Docker container, and this is something that's updated up to this day, that features all of the programs in OpenWorm. So this is a little bit, I don't know if you're going to be able to run this or not on your computers, but there's a Docker container you can download from our GitHub, from the OpenWorm GitHub, and that OpenWorm GitHub link has this Docker container, and you open it up on your machine. And I think the best way to do it is to use a Linux machine, but I think it can also run on Windows. I'm not sure, but it basically opens up and it runs all the simulations. So there's a biophysics simulation, there's a connectome simulation, there's a simulation of the cells and the neurons. There are a couple other simulations in there, and those will all run as you run this Docker container. And it generates output files like graphs and images. So it's really supposed to be instructive of the whole project and like kind of the whole scope of the worm. Now, DevoWorm has never been able to put anything in this Docker container, unfortunately, so we're not represented in that. But I've tried to get in the past people to contribute something. I've always wanted to build like a three-dimensional model of the embryo, but we've never really gotten there. Something that was suitable for that. So that's, it's probably not what you're doing in your projects, but, we can give it some thought as to maybe how to, maybe later in the project period when we're, working on the projects, how we might use some of these things later. And so it may be that we make something that will apply to that Docker container. So any questions at this point?
Yeah, that sounds like a good idea.
Yeah.
The Docker container with the 3D model, right?
Yeah. It's just basically,
At least for hosting, I think, like we could explore it as an option, like as an additional option, because then things will be in our hand rather than giving Heroku using Heroku as a platform. It has its own limitations with the file sizes and the extent or the number of requests you can have per month. So,
Yeah. So any other questions? Okay. So that's all I'm going to talk about with that. Then I want to go over to some of the other resources that we have here. So we have so some of the data sets. I know I sent a link to Jiahang and Wataru about some of the data sets they might use for the graph neural networks. This paper here, which is the DevoLearn paper, this is something that we wrote is sort of a technical paper for the DevoLearn release. So this is where we're using deep learning to analyze microscopy data. So here's some microscopy data that was analyzed, and basically the embryo is segmented, and you get these different outputs of defined cells and their morphology. And it's a pre-trained model, so it's something that we've had to use data sets to benchmark. And one of the things I wanted to point out here is that there's a table in this paper. It's table 1, and this table has the different data sets. So these are different data sets that are available online. You can download them and use them to benchmark your models. So this is, like, for example, segmenting the nucleus in C. elegans embryo. This is from a cell tracking challenge. This is a good data set for looking at nuclei and their position in the embryo. So this is something that has data that basically features a fluorescent nucleus or a fluorescent centroid for each cell over time, and that gives you a data set of points that you can then generate, distances, and you can generate a graph from that. There's also segmenting the cell membrane. There's this data set from 3D MMS, which is it's a morphological paper on morphological segmentation of the embryo, but it might be useful for other types of data that you might want to use. So these are all data sets that are available publicly. You just download them. And there's another source that I'll send you. It's the systems biology database, which has some other embryo data sets in it as well. I'll have to send that to you guys after the meeting. But this is another database that we use. It's run out of Riken in Japan, and they have a number of different data sets that are usually images like this that you can then take and find features on it and then use those features as input data. So there's a lot of data online for this. We have something called the DevoZoo, where we have short microscopy movies. We have these SPIM movies in particular. So these are secondary data sets. This is actually not a SPIM data set, but this is another data set that you'll find useful. It's like microscopy. This is SPIM, which is a certain type of microscopy, which is very high resolution. So in both of these data sets, you have microscopy images of the cell. You have markers inside of the cell that are fluorescent. They mark different features in the cell, and then you can take that and extract the data as you wish. In previous years, we've had people take videos. You extract the images frame by frame, and then you put those images in an algorithm, and you do some segmentation, some basic segmentation to get a general idea of where the different features are in the embryo. And then if you're doing something like graph neural networks, then you need to get the data, you need to build a reference frame, and then you can build these networks, because you need to know kind of where they are in space, but also where they are in time, because your embryo is, the number of cells in the embryo is growing over time. So that number of cells is going to increase. The reference frame is going to change. You need to normalize that spatial reference frame. You need to keep track of the time, So each frame in the movie is a time point in development. And then, you can build a model from those two pieces of information. I guess it's 2 dimensions of space in a lot of these movies, because you don't have a microscopy stack. This is all just that one frame, focal frame. So it's just 2 dimensions, but then you have this third dimension of time. And then from that you can build actually a 3 dimensions of space, because we know that the cells have a certain z axis location. But that's something that we have, we can label that after the fact, because we have that data in some of these other data sets. So there's a cell position data set here that has XYZ normalized position for these cells. So, this is something we can work out over, maybe we should start doing this now, in fact, figuring out these coordinate systems and how to get the cells, arranged or organized. It shouldn't be too hard, but it's going to take some playing around with the data. I just wanted to point out, though, that we have, I think, all the data sets you'll need to do this. So we just need to kind of go back and forth on how this is going to look. Then I wanted to bring your attention to the GitHub repos. So I've had all of you join our DevoWorm GitHub organization. So this is DevoWorm. It's not actually an organization, it's an account, but it's fine because it basically gives you permissions for this repository. So this is the GSoC 2022 repository. Here are the directories that I want you to use for pushing your work, maybe weekly. If you have work on your own repository, if you could clone it to this, that would be good. Because you have permissions now, you don't need to issue a pull request. You can just push it up to this place. However, it's nice to be able to review pull requests if there's something that you need me to review. So keep that in mind. I want you to update these maybe once a week. Basically, anytime you do work. And I know you all use GitHub, so that's not a problem. But I want to basically make sure that people are doing their work and basically having it backed up and having it in a place where we can evaluate it. So these are public repositories, and I've given each project its own directory. So DevoGraph, which is Jiahang, you push your things to DevoGraph. GNN's developmental networks would be Wataru. And then the digital microsphere, you would have digital microsphere, where I think would be Harikrishna, and modeling axolotl embryos would be Karan. I don't know, maybe that's, I don't know if you want to stake claim to one directory or another. That doesn't really matter. I just went by the title of your proposal. So this is something that, if you want to be able to, I just want to make sure that your work is in separate directories, that's all. So whoever makes the first push to whatever directory you make your push to, the other person will pick the other one. And then in the README, maybe we can make that clear as to who's pushing to what directory. Just so we can keep everyone's work separate and I can evaluate it and we can go through it. But yeah, so then we'll have this as a historical repository, and then after, towards the end of the coding period, we'll do like a review of work, and then we'll, try to work on getting it. Google wants you to have something that you submit to them that has some sort of executable in it. It could be an executable file. I think it could be a Docker container. It could be a set of digital notebooks, but it's got to be something that they can run and see that it works. So that's what you have to aim for at the end, and all of that would be do it in this place in GSoC 2022 in this repository, and then I also want to at the end of the coding period push it to the DevoLearn organization, which is a formal organization, and this is going to be part of DevoLearn as well. But I wanted to do the development here. I just wanted to give you a heads up on that. So, okay, so Jiahang has a question. Hi, Bradley. Could I use some time in the meeting to introduce some resources related to GNN and biological analysis? Yes. So go ahead, Jiahang.
Oh, yeah. Okay, let me share my screen.
Okay.
One moment. Oh, probably I have to quit the meeting, and I will come back again.
Okay.
Yeah.
All right.
Just wait a moment. Yeah.
All right, that's good. All right, did we have any other questions while we wait? Okay. After Jiahang's done, I'm going to go through some of these readings on, well, I'm going to just go really quickly over the open source readings, and I encourage you to explore that repository, as it has a lot of interesting things on like, how to manage open source software. Once you create open source software, then what? And that's really the question, because there's Jiahang, he's back.
Bradley, I'm working on trying to get a grid on some spheres.
Okay.
I have a couple of ideas, so I'm working on that for Harikrishna and Karan.
Yeah.
Well, I'll keep working on it.
Okay. Yeah, that's great. Yeah, they were just mentioning that they were working on some other materials for that. So yeah, if you guys want to email each other on that, that'd be good. So Jiahang, were you ready to go?
Yeah.
Okay.
Oh, there are some issues. I cannot share my screen. I would just. Sorry, that's.
If you share the tab instead of the whole screen, it'll work.
Yeah, it didn't work.
Oh. Do you have a link to these
Oh.
materials, or I guess not?
Yes, I
I do.
will share these links in the chat.
Okay.
Yeah,
Yeah, I'll bring it up here.
Yeah. And I've surveyed some resources this week. Yes, it's about the graph neural network and biological analysis. Yeah, and first of all, I want to introduce some conference because I remember that you mentioned the learning on graph, right?
Yeah.
Several weeks ago.
Yeah.
Yeah, and yeah. It is a new conference which is especially for the graph neural network and graph machine learning. But actually, there are some other cool conferences for the GNN and its application, such as the iClear
Yeah.
and Triple AI. And this, I remember that the deadlines of these two conferences will be in this year. It will be probably the October and November. I don't remember exactly, but yes, I think we still have time to prepare for these three conferences. Yes, and actually, such as this one and this one, and there are some other more conferences for our work in the next year. Yes,
Yeah.
and yeah, this is the first part. I mean, we have a lot of conferences we can consider if we want to submit some works to these conferences. Yes.
Yeah.
And also there are some other things such. I want to introduce the second part is about graph neural network and its application on biological analysis. Yes. And the second part is I've surveyed some papers, and I will put these names in the chat.
All right.
The first one is graph neural network for cell tracking in microscopy videos. It is probably related to our work, yes, because we are focusing on the developmental analysis of embryos. Yes, and actually, this paper is trying to introduce how to use dynamical temporal GCN to analyze or track the cells, such as they use GCN the graph convolutional network to aggregate information for each, each snapshots of a video, and they use such as the recurrent neural networks or LSTM to aggregate information across different times or different snapshots. Yes, and I remember this. It is a work published in the February this year. Yes, and it is a very new work. And I didn't see other works regarding using graph neural network for cell tracking. Yeah, and another work is this one. This paper is utilized. This work is utilized to solve some issues regarding the traffic prediction. But actually, I think the traffic prediction is very similar to the cell tracking. Actually, if you have delved into this paper, and it is very similar to the graph neural network for cell tracking. I mean the TGCN, and I think it is they are very similar, so I will not introduce the D-GNN, but I think maybe you would like to take a view of this paper. Yeah, it is very interesting. Yeah.
Okay.
Yeah. And there is another one. An inductive representation learning on temporal graphs. I mean, these three papers are using dynamic or temporal graph neural networks on some application stuff. And the third one is using the GAT graph attention networks, which is merged to the transformers. Yes, and it is very another interesting.
Yeah.
I want to introduce another work which is called this one geometric graph convolutional networks. Yes, I'm not sure this paper is related to our work. Yes, because it is a work try to how to say this, project the discrete space of a graph into the latent continuous space, such as the hyperbolic space, such as other spaces. And this paper thinks we can find, more useful neighbors or more advantage neighbors from the latent space. Yes, and yes, I think it is interesting, but I'm not sure it is very related to our work. Yes.
Well, yeah, I think it is actually.
Yes, the first.
Yeah.
Yeah. Go ahead. You.
Oh yeah, I mean that's one possibility. You know, once you build a network, it's not. It's kind of like you're creating this normalized space because the data that you're going to get is over maybe several embryos, or you're going to have a lot of displacement in the images because it's kind of things are moving around. So one way to do that is to create sort of a normalized space, a metric space of some type. And so, thank you for attending, Harikrishna. And so one way to do that is to just do like a mathematical function to transform the points to a centroid. Another thing is to map it to some higher dimensional space, and that would be like hyperbolic space is often used in network theory actually to do this, especially with something that has a curvature like an embryo, where you need to sort of approximate that curvature, but you don't want to necessarily put it like on a sphere or something. I mean, you can put it in a space where you have that accounted for.
Yeah. Actually, I think, you're right. But, do you think it is related to our work? Using the geometry stuff?
Yeah, I think definitely, because you do have some inherent geometry here that, like, with Karan and Harikrishna's project, they're taking an embryo and they're creating a sphere. They're taking flat images and they're putting it on the surface of a sphere. In microscopy images, they're two-dimensional. But like I said, when you collect microscopy images, it's a stack of images, and every cell you observe in that focal plane that you have in your movie, which is two-dimensional, has some depth. So that image has depth. It's just that you're not seeing it because you're at different focal lengths from that cell. So you go down, or you're looking down at it, and you'll see like these different. It's like if you look down at a city and you see the rooftops and you see the buildings, they're not all the same height. They have different heights. So the rooftops are different locations in the z-axis. So having that sort of geometry, you can actually have a model of, or you can project some of those that Z information into your representation. So definitely it is useful.
Yeah, okay, I got your points. Yes, I think I will survey more papers regarding the geometric graph neural networks. Yes, there are lots of related analysis. Yes. Yeah. And I will read another paper. Yeah, you have introduced this one to me. Remember that? This one. Yeah. Self-differentiation processes of spatial networks. Yes, I've read this paper. Yes, and I think And I think you are trying to utilize the tools of network to analyze the developmental process of embryos.
Yes.
Right?
Yes.
And such as the diffusion of cells, such as the clique, or such as the cluster. Yes. So I think because I'm trying to, because in my proposal, I'm supposed to build kind of that can, I think they can build a graph based on the videos of embryos. Yes.
Yes.
So our target is to get the graph. I mean, the graph should incorporate some useful information that can be utilized by the network analysis tools. Am I right?
Yes.
If yes. So it is about how to build a good graph, a good data set of graphs that can incorporate the useful information of embryos, such as the developmental process of these embryos. I think this is a target of our projects, the GSoC 2022.
Yeah.
Right?
Yeah.
Yeah. Okay. Yes. I mean, yes. I remember that there are some other papers. Please, wait a moment. Such as this one.
Oh, right. Yeah.
And this one. Yes.
Yeah.
Yeah. Actually, I checked these papers as well, and I found that there are some work. There are a lot of work trying to use the network tools to analyze the developmental process of embryos. Yes.
Yeah.
So I think it is very important to build a network so that you can use these network analyze tools to analyze the network itself.
Right.
Yeah.
Yeah.
So So I think there's a lot of targets of the GNN projects in the GSoC. Yeah.
Yeah.
And I'm done. This all the parts I want to share today.
Okay, well, yeah, thank you. That's very good. Very thoughtful, going through all these different resources. And I definitely think, yeah, so, I mean, one of the things, of course, we want to have is that geometric aspect. Then also the temporal aspect, which is, because you have things in time that are unfolding. So, I mean, there's a lot of, I guess, this is a very hot area. There are a lot of papers coming out every day, every week. And certainly, it's something that, now with the conferences, oftentimes you'll have people doing like technical work on some technique. And so if we're doing stuff with more applicable to biology, it depends on how innovative the actual GNN is. That depends on whether we get accepted or not. That being said, that doesn't mean that it won't be accepted to some of these conferences, because it is competitive. So, get it, like if we're jumping ahead here, but if we're thinking of submitting it somewhere, that submission has got to figure out how to make it so that they really see the value. And it's not that it doesn't have value in it; it's that these different venues have different sort of they have different priorities or criterion for what they want to see in their top papers. So I mean, we can borrow a lot from these papers and then apply it to biology. And it may be that, we even target like maybe something like a computational biology conference or journal as well. That might actually even be a better fit. It depends on how much progress we make in the coding period. And maybe, if you would like to, I know that Karan and Harikrishna are working together. Maybe you could get in touch with Wataru in the Slack and see where he is. Because there's a lot of, I want to make sure that you guys aren't doing the same thing and doing the same work.
Yeah.
Yeah.
Yeah. Yeah. I tried to contact him. Yes.
Okay.
Yeah, we didn't chat yet.
Yeah. But I think with something like this, it's like, we want to have like complementary work maybe is the most powerful. So if there's something complementary you two can do, that I think then, when it comes time for a paper, we really have like, a lot of ground that's been covered, and it'll make for a stronger set of things that we can produce from this.
Yeah.
Okay, now I'm going to go over a couple of the readings that Jiahang couldn't share on his screen, and I'm going to go over one more thing in the reading queue. So let me go to share my screen here, and I will talk about some of these papers a little bit in brief. So the first one he mentioned was Graph Neural Network for Cell Tracking and Microscopy Videos. This is on the archive. So this is where they present a novel graph neural network approach to use for high throughput microscopy videos. So this is a nice paper that kind of talks about how to extract data from microscopy videos and build a basic cell neural network or graph neural network from it. So this is nice because it focuses on lineage trees, which is exactly what we're trying to do in our project. So the next one is TGCN, which is a temporal graph convolution network for traffic prediction, and this is for traffic forecasting in real time. This involves integrating a number of systems such as urban traffic planning, traffic management, traffic control. So this is something that shows sort of the predictive power of these networks. So a TGCN is a temporal graph convolutional network model. So this combines graph convolutional networks and gated recurrent units. So this has a specific type of topology, and we'll see if this is something that might be of use. This gives you spatiotemporal correlations. So this is something we're looking for in our developmental data. The next one is GCN, which is geometric graph convolutional networks. This one is based on, it's sort of specialized for geometric features. The basic idea is that the aggregation on a graph can benefit from a continuous space underlying the graph. So again, we need our coordinate system for our embryo data, and this is a continuous space that's built under the graph that gives you a coordinate system and then spatial relationships or geometric relationships. And of course, as we mentioned, you can have different spatial frameworks and other things that will, the sort of latent space that allows you to reconfigure your representation. Finally, there's this inductive representation learning in temporal graphs. This deals specifically with the temporal aspect of what we want to do, and temporal graphs more generally. Our lineage trees are actually examples of temporal graphs, although they're not really what they're talking about here. Our data is basically in that sort of orientation, a temporal orientation. So they propose that this approach called temporal graph attention, or TGAT, can be used to efficiently aggregate temporal topological neighborhood features, as well as to learn the time feature interactions. So temporal topological neighborhoods are basically areas of like geometric areas, topological areas of the images or of the data set that emerge in time. And so this is something that's very relevant to what we're trying to do. So this is just, they use this attention mechanism to sort of develop novel approaches, functional time encoding approaches to this problem. And so this might be very useful as well. Okay, now we're going to go to one set of readings that I wanted to talk about, and this is a very interesting set of research from, I think it's from a single group. I can't remember, but let's look at this. So this is about parasite collectives, and you're like, why are parasite collectives interesting to development? And the answer is because they form very interesting patterns, and we'll see some examples of those patterns in a minute. So this is one of these sort of profile articles that you find in journals. This is out of I believe Nature Physics, and this is called "The Power of Parasite Collectives." And so they're talking about these organisms called Plasmodium sporozoites. And so these are different. These are Plasmodium that can move in rotating vortices owing to their chiral shape and mechanical flexibility. So a chiral shape is like a spiral. So like if you think of corkscrew pasta, that's sort of a chiral shape. It has a direction that turns, and it turns upward or downward depending on how you're moving along the chirality of the shape. Of course, DNA is also chiral, and we know that there's a handedness to DNA, so it means that there's a right-handedness to DNA, and also amino acids have this same sort of chirality. There's a curvature, and the curvature has to be in a certain direction in order for it to be biologically viable. So these chiral shapes are important in biology. Mechanical flexibility is also an interesting property. We've talked about soft materials in the group before. And these things taken together reveal important physical aspects of collective motion. So we've talked a lot about collective motion in the group, about swarms and other sort of collective behaviors. In this case, they're talking about parasites that behave in these collectives. And the properties of the parasites themselves actually lead to these sort of biophysics. So to perpetuate itself, the tiny micrometer-sized parasitic microbe named Plasmodium has evolved a strategy of colonizing and proliferating in two distinct animal types, namely mosquitoes and vertebrates. So they're parasites that exist in a wide variety, or in two different types of organisms that are vastly different in terms of their physiology and their biology. Whereas the mosquitoes' reproductive fitness and life expectancy are barely affected by these parasites, humans pay an enormous price, as we know from human history. The transmission of Plasmodium sporocytes rely on their ability to transition from their mosquito to their vertebrate hosts. So they're in the mosquito eats vertebrate blood, they're infecting the mosquito, and then they get transmitted to a vertebrate. And so they move between these two different types of organisms, these two different groups, quite effortlessly. So remarkably, only a few tens of sporozoites are inoculated individually, along with the saliva fluid of the mosquito. So they basically have a very small founder population that moves into the host or into the infected individual, and then they proliferate from there. And so they enter the blood capillaries and continue to the liver. So there's this other paper in Nature Physics, a full paper, where they carefully examine how the mechanical properties of these parasites can profoundly influence their individual and collective dynamics. So once they're in a group of individuals, they can exhibit these different dynamics as they go through the bloodstream, and that determines sort of how they propagate and how they move. Microscopy of individual sporozoites gliding through the elastic dermal layer. So they're getting through the skin, and they have to make it through the skin, and then they have to make it into the bloodstream and move through the bloodstream. This has revealed a striking flexibility. Yet little attention has been given to their mechanical properties. By squeezing high concentrations of sporozoites from mosquito salivary glands onto glass cover slips, so they are able to harvest a large number of these sporozoites onto a cover slip. So they're doing it, taking it out of the mosquito and putting it on, and under the microscope. The authors have designed a clever ex vivo setting to address this unsolved issue, so they can actually observe these dynamics. You'll put like on your microscopy cover slip, you'll put some buffer, and then you'll drop these in, and you'll be able to observe them moving around in the liquid. Under these conditions, the authors find that aggregated sporozoites form rotating vortices, in line with the circling patterns of Plasmodium previously imaged in salivary glands. So they can image these things in vivo, but you can see these ex vivo as well, or under a microscope. Looking at several vortices at once, one can see that the tightly aligned crescent-shaped sporocytes always rotate in the same front-first direction around a common center, an example of chiral collective motion. So they have this chirality to the motion. It's going, in sort of this swarm, but the swarm has a very distinct structure around a central point. These vortices are reminiscent of other spectacular collective motions observed in certain animals and bacteria. Rotating vortices often arise when these move towards an attractant, such as a food supply or pheromone trails, while avoiding collisions with neighbors. So there are a couple of very simple rules here. The first is that you have, this sort of taxic behavior, so it's being attracted to something or repulsed from something, some environmental stimulus. Then you can't be too close to your neighbor, you can't be too far away from your neighbor. So you have that. If you're familiar with the Boyd's model, this is exactly the type of thing that you see there. You see these very simple rules, and then you get this sort of complex collective phenotype that emerges from it. A variety of mechanisms and fitness strategies have been associated with rotating vortices and reflect the huge diversity of vortex scales and samples. So this is an example here of what we're talking about. So this is the key ingredient of vortex formation. So you see these Plasmodium sporocytes, they're bent and they move in a certain direction. They have static interactions, so they're repulsed from one another if they touch. And then they have this mechanical flexibility. So you have this thing that can bend. You have this rod-shaped thing that can bend. It moves in a certain direction. It just turns along the bend. It can't touch one, so they're repulsed by one another if they touch. And then those three things together form these vortices. And you can see a vortex of fish at the left, a vortex of sporozoites in the middle, and then a self-sorting example with the same phenotype on the right. And so you can see this is sort of a control for a self-sorting phenotype. This is a vortex of fish, and then the vortex of sporozoites is sort of in between there. And so that's an example from this one parasite. Individual sporozoites glide on a substrate in a circle owing to their curved shape. They display a strong preference for the anti-clockwise direction because the chiral shape of the sporozoite's microtubule cage dictates the orientation of the secretory organelles and proteins necessary for adhesion and motility. So this is all in the service of them moving more swiftly through the medium that they're trying to get through. And so the paper itself here is also in Nature Physics. And they talk a little bit more about this. They do some agent-based computer simulations. They do image analysis. They quantitate that image analysis, and they build the simulations from the data. And they basically make the same observations, that the mechanical flexibility allows single sporozoites to be sorted according to their curvatures and speeds, and that these effects increase with vortex size. So the vortex size is limited by the phenotype and the way that the phenotype behaves in the collective. We also find that the vortices undergo oscillatory breathing, which is an interesting point, which means that it's pulsating in some way, because the thrust from the motility force of the single sporozoites can be stored in their elastic energy. So this is interesting. There's motility going on. There's each sporozoite has an elastic potential, energy potential, and this results in this sort of breathing of the collective. If you think about, like, if you've ever watched schools of fish in coral reefs, if you've seen video of that, or if you've even seen it up close, you see that there's a sort of pulsating, and the collectives aren't just these static clouds that move. They kind of pulsate, and they have these internal dynamics. So that's what they're getting at here. In general, our work demonstrates how single particle shape and mechanics can determine the dynamics of large active collectives. So this is an example here, a little bit more about some of these collectives and how they're rotating. So this is an example of how you might get an infection of these parasites. You have the mosquito, and the mosquito bites a human, say, and these parasites go into the bloodstream, and they go through the salivary gland, and they have to go through the skin of the host, and then they get into the blood vessel, and they move through the blood vessels. And so you have this medium of elastic material, which is the skin, viscoelastic material, and then you have the bloodstream, which is also viscoelastic, but less dense. And so they're moving through that. And so to do that, they have to have this sort of adaptability at the collective behavior level. And so here are some examples of different types of observations that they've made. You can see the single phenotype of a single parasite here, a sporozoite, and then the collective sporozoites here, where you have a bunch of them moving around in a synchronous fashion. So they kind of go through some of the stuff. They run their agent-based simulations on this, and so they show some of the examples of the physics and how they affect this collective. And they don't have any good pictures of it, but I think this is the model sporozoite, which is where they model each. They model these chains with a ball and spring model, which is something that's very common in biophysics, where you think of the individual units of this chain as individual sort of nodes or balls, and the balls are connected by this link with a spring, so it's elastic, and then it can move back and forth. So there's a lot of potential motion within this chain, but also the chain itself moving. So this is a sporozoite phenotype modeled as a ball and chain system. And so you can have this, you can measure things like the force distributions and the elasticity using this type of model. You also have these different radial distances, and you can measure the behavior of the collective itself. You can look at the radius of curvature and how it affects these different collectives, and you can also look at the speed of the rotation. You can look at the collectives. So this is all done in simulation. This is taken from measurements, but the actual biophysics and these graphs are generated from a simulation. So then this is more simulation data. The simulations predict self-sorting and rotating vortices, so they're able to replicate that finding. And then you see these vortex size fluctuations, so they're not static. They don't just kind of sit in a deterministic, swarm or whatever you want to call it, collective, but there's a pulsating along the edges, and sometimes they break apart, I imagine, because that's what collectives can do sometimes. And so you can see the different shapes here over time of these collectives. Okay, so that's pretty much all I wanted to talk about the papers. This is something that was put out on social media. This is a picture from this work where they say, we then used agent-based modeling to identify the required physics elements. In particular, his analysis, the authors, demonstrated that the propulsion of the single sporozoites is converted into elastic energy of the vortices, making them a model system for active elastic matter. So this is something, and we've talked about in the context of embryos, but you see another example of active elastic matter being these collectives of very, very simple organisms. And here you have this bridge between experiments and models was quantitative image processing. So this is something that, you put these things under a microscope, you look at the collective under a microscope, and you can actually track the cells, you can track the sporozoites in their sort of active state as they're moving around, and you can track them and then use those data to build the simulations. So the microscopy was really the linchpin of all of this, to bring this to something that we could actually simulate and measure, for that matter. And this is able to show that the sporozoites and the vortices are sorted according to speed and curvature that the vortices undergo during their shape oscillations, or that results in shape oscillations of the collective. So that's all I wanted to talk about with those papers. Thank you for joining me. Okay, so I'm going to finish up here with, I'm going to go over this repository a little bit of the different papers, and Harikrishna will be able to see this later, so will Wataru. But this is the open source and open science reading repository that I posted in the Slack. And it has like some books, like there's some digital books in here, which are, really for your reference. I don't mean for you to read them cover to cover, but I just want you to have this as a reference. And then these folders that have, different topics that you might be interested in. And then this is a laboratory contribution philosophy, which is maybe less relevant to you, but is maybe interesting if you want to look through it. This book is a particular interest to maybe, when you're thinking about long-term about your projects and what you're producing. So this book is Producing Open Source Software, How to Run a Successful Free Software Project. This is Carl Fogel, who's a developer. And so, there have been a lot of open source projects. A lot of these projects fail, but some of them are very successful. So if you think about like Linux, or you think about some of the other, like, Firefox or other types of free and open software, they have, have to have successful structures to run their contributions. And so in OpenWorm, for example, we've had no problem attracting people into the project. Like, people always come by the Slack and check it out, but it's, it's a little harder to turn that into, contributors who are producing something of value. So that's why we run GSoC, for example, because it brings people in and it gives them a project to work on. But this is something that all projects face. And when you produce open source software, there are a lot of things you need to keep in mind that are important for making it successful. And I mean, not just short term, but long term as well. So this book has a lot of, and this again is for your reference. You don't need to read it cover to cover, but there are a lot of things in here. So, there's the history of open source software, sort of the difference between free and proprietary software, free versus open source. So if you're open sourcing something, it's not, locking it away. You might have the Windows operating system is proprietary. You can't freely modify it. You can't freely distribute it. But with open source software, both of those things are true. Now, open source isn't necessarily the same thing as free. You can have something where you open up the code to people, but it's definitely not free in the sense that you distribute an executable. I mean, there are different ways that people do this. There are open source organizations that have software that isn't quote-unquote free. There's free software that's open source. There's free software that isn't open source. And then, so that's something to keep in mind when you think about licensing your software. Now, we have in this group, we tend to stick to things like the MIT license, which is an open source license that, enables different people to do different things with the software. And it sort of, keeps people from using it in different ways that might not be what we would want to see in the world. So, there are a lot of different things you need to think about when you work on projects. You maybe think of a good name, something you won't necessarily have to worry about, but we work on with in the DevoLearn area is version control and versioning. So how do you make a version of something, a new version of something? So these are all things that you might learn about and apply to your work later in life, or we can talk about this with respect to DevoLearn. And there are all sorts of different things like choosing a license. There are all sorts of open source licenses. There's also discussing things, having discussion groups surrounding your software. How do you make that productive? You need technical infrastructure. You need social and political infrastructure. So, there's in a lot of open source projects, there are different types of leadership. You have benevolent dictator types of leadership. You have totally democratic types of leadership, consensus-based democracies, and this, shapes how these open source projects actually come to fruition. If, sometimes the benevolent dictator model is good for driving forward innovation, but not so good for, managing changes. There are other ways that you can make money off of open source. So there's a whole section on economics of open source and how those things get financed. If you're interested in open source as a career, a lot of companies are looking for open source people or people who know open source. And a lot of companies are investing in open source, not necessarily on their own, but to use those tools in different ways. So there are different things, to keep in mind long term about what you're learning in this program. Then there's communications, which involve like how do you communicate to people what you've done. We definitely are going to be writing up the results of our projects for submission to Google, so this is maybe something to pay attention to. There's managing participants. How do you get people to say, how do you get a maintainer, for example? How do you get people to maintain the platform so that it stays current? It's one of the biggest challenges we've had with DevoWorm is that, time goes on after these things get released. And as time goes on, especially in machine learning and in deep learning, the pace of technology is very fast. And so people need to be able to manage these fast changes and maybe, contributors coming in and wanting to add on security features or newer features that are, modern. And by modern, I mean a year later, it's modern as opposed to what you have, which is growing out of date. So these are things you need to think about when you release open source software into the world. How does it get maintained? And so, and then of course, you have people who just contribute. How do you get contributors to come in and contribute to your software instead of just you? We've done things like Hacktoberfests, which are organized things that GitHub is involved with. And these sorts of activities drive people to, contribute, and they reward them for contributing. And there are a lot of ways to get people involved in your open source platforms and how to get it maintained and how to keep it relevant and all these things. So I just wanted to bring that to your attention because I think that's important to think about long term in terms of what you're learning here and what you might do in your career. So, any questions about that? Okay. Well, if that's all the questions, we can ask questions later in Slack or via email. But I think we'll wrap it up for today, and I hope that you have a good week. I know for the GSoC students, I'll be continuing to give you information in the Slack. I'll probably give you some more things for Community Period. And again, I encourage you to go into the OpenWorm organization, OpenWorm Foundation, and learn more about what they're doing. That might be useful. Otherwise, let's just keep working on getting our projects ramped up. And hopefully by when the coding period starts, we'll be able to hit the ground running on things and get our, stick to our schedules. Okay. All right. Thanks for attending. Have a good week.
Well, bye, Bradley.
Bye.
Bye.
Bye. Bye.